Okay, we're gonna get the fireside chat started. Just a few words for why and how this got started. And as we were planning this last year, we thought, okay, we want to finish with a bang, and we wanted, but then we were faced with a challenge of what to put on to end the show that can top what you just heard throughout the day. So we thought we had to get someone on the stage. This person probably should not be a Stanford faculty again, and but we thought, who are the most important people that we can think about that has been so instrumental to the institute, but is not a Stanford faculty. So we came up with Clara and Cori. And Clara is the executive director of the Clara and Joe Tsai Foundation. And Clara graduated from Stanford with an MBA degree and went on with to have a very successful career in the business world. 5 years ago, we had the opportunity to rename our institute in honor of her foundational commitment to Stanford neuroscience. Thank you. She's been a very thoughtful partner for Bill Newsom in the early days of Institute, and has been a great experience working with you in the last year as I was stepping into this role, and I look forward to continuing working with you. And Cori Bargmann is the Lulu Anthony Wong Professor from Rockefeller University, and she is a renowned neurobiologist, internationally renowned neurobiologist, I don't have to tell you. Her lab studies how genes and environment generate a variety of behavior using nematode C. elegans as the model system. Cori has studied this question, has really put olfactory system of C. elegans on the map, and has studied neuroscience at the level of genes, cells, neurocircuits, behavior, and evolution, and has trained generations of scientists, myself included. Thank you, Cori. I think Cori really is the encyclopedia of neuroscience. And for anyone who knows her, you'll believe me, you'll agree with me that her knowledge is really far-reaching and into every corner of neuroscience. And it's an incredible honor to have you here with us today. And Cori has also been very much affiliated with us, and she was on Bill's kitchen cabinet, as Bill called it, and came for the retreat as a keynote speaker, and will conduct our external review next year. Thank you, Cori, again. So I have a long version of Cori's introduction that can take 10 minutes, which I won't do now, but that's right. So please join me in welcoming Cori and Clara to the stage.
So we're just going to have a conversation about some questions that the two of us have been talking about and emailing about as we got ready for this event. I just want to start, actually, because I am a guest here, by saying how glad I am to be included and what a joy it's been vicariously to be involved in this institute since its very beginning. Bill Newsom and I were working together on the BRAIN Initiative when this got started, and so we just sort of used this as a way of continuing our scientific relationship. And every time I'm invited to Stanford, I very quickly say yes, and just to have more opportunities to hang out with this group of people. So thanks a lot. And Clara, how did you get interested in neuroscience? What is it that brought you to this in your?
Well, first of all, it's such a dream for me to be on a stage with Cori Bargmann, and so I think I'm going to savor this moment for one thing. But yeah, so I think originally I had been involved in having a lot of conversations in general about interdisciplinary science at Stanford. But then I started to have some deeper conversations about the brain and understanding just the remarkable adaptability, the adaptive capabilities of the brain, or neuroplasticity, and was really impressed with that. Had a lot of conversations with Carla Schatz. She was actually a really important piece of drawing me into neuroscience here. And so, that really sparked my interest in neuroscience. Then I started to learn about Bill Newsom's efforts to shape President Obama's BRAIN Initiative, which you were part of, and also to shape the Neuroscience Institute here. And so I kind of figured that it was like a pivotal moment in bioscience, and I decided, okay, I was going to act on this opportunity and help to bring Stanford neuroscience into its fullest potential. And I have to say that one of the, I think, lofty goals of the Institute is to understand the mind in all of its human complexity. And I really love that. I think it's really motivational, because not only is the mind the basis for society, but it's also the foundation of everything at Stanford, across all of the disciplines, arts and science, humanities. So when you invest in Stanford neuroscience, you're not only role modeling interdisciplinary science, but you're really lifting up all of Stanford scholarship. But Cori, you were also a part of the BRAIN Initiative, and you've been involved in many, many different groups that have been studying neuroscience. And I was wondering, when you started with the BRAIN Initiative, what did you think was the potential of brain science? And how, what have we advanced in the last decade? And what do you think is the biggest advance that we've had in brain science over the last 10 years?
So 10 years is a good amount of time in science. In 1 year, it seems like almost nothing has happened. And then in 10 years, you can look back and really see how fields have moved forward. And I just want to pick out one example of something that I think the BRAIN Initiative decided early on to do. And that was to come up with a parts list of the human brain now, and specifically to identify all the different cell types within the brain and how many of them there were and who their neighbors were and what genes were active in each cell type. And that sounds like a kind of boring cataloging problem. But on the other hand, if you don't have the parts list, you don't know what it is you're trying to study. And second, once that existed, it enabled a huge amount of additional science to take place, true discovery science. And so, for example, the first thing that happened was that people looked in the brain and realized that there were many, many more distinct cell types with different patterns of gene expression than they had realized. Things that they had thought of as sort of like regions turned out to be just like incredibly interspersed, complex environments with many different molecular compositions. So that immediately posed questions like, what are all these cells doing? And then second, it gave you basically a barcode or a handle that you could use to get into any of those cell types and ask what those cells were doing. So these cells could be labeled. You could figure out where they projected to, where they projected back to. Turns out that the connections between different brain regions were quite a bit more complex and interspersed than people had realized. You could use the sort of modern tools of optogenetics that were largely developed here at Stanford and other related pathways to figure out which cell types were doing what. And now every time I pick up a journal, I'm kind of astonished by the amount of information we've developed in understanding. And that just is almost, like in the background of what people say. It's like, oh, the entorhinal cortex and the hippocampus are mutually connected with each other. And that now has really a precise composition statement. You can go in there, you can ask what each of those two things are doing. And that's been a really spectacular advance for the field. So it's really been leading to a lot of discovery, particularly for regions that are not the cortex, which neuroscience has studied for a long time, but regions deeper in the brain, which are very heavily involved in motivation and emotion and learning and physiological functions, had just not been studied in the same way. So I just think in terms of discovery, we now see what the brain is in a much more detailed way, and the problems are set out in front of us in a big way. Now that's the discovery side. The second really important thing that came out of that parts list is that it enabled us to connect human genetics of disease to brain function in a new way. And specifically, shortly before this work was done, people had really gone big into this problem: what's the genetic risk underlying different human diseases, specifically neurological and psychiatric diseases. Huge studies of genetics, huge identification of risk genes. And for example, a really important thing that came out of that was the recognition that most of the risk genes associated with Alzheimer's disease are not expressed in neurons. Most of them, actually the highest genetic risk, is associated with microglia, the brain's immune cells. And so that has completely reformulated the way that people are thinking about the causes of Alzheimer's disease, not purely about the cells that die, but in the environment in which they find themselves. And actually, one of the winners of the postdoc prize this morning was actually working specifically on TREM2 and trying to replace that within microglia, which is exactly one of the things that has come out of that new insight. And so that idea of like being able to look at that and kind of just reframe your idea of what the problem is that you're trying to solve and link that to human disease, I think has been really a significant advance for the field. So those are just, there's still plenty of new things to know, but I think those are things where you can kind of look at something that we just did not know now, and that's moved us both in science and in medicine in the direction that we want to be.
That's great. So I'm interested, you are in a unique position because you've been involved in so many different collaborations internationally and nationally to move brain science forward, including CZI, the Brain Initiative, and many others. And here at our institute, our strength is so many great disciplines in close proximity. But I was wondering if you see other pathways, other ways to do neuroscience that might be just as effective.
So I think the important thing to realize in science is that no one kind of institution or funding mechanism or scientific mechanism has all the answers. And it's to really figure out what you're good at and to do that well. So one thing, a lot of people like to complain about the federal government. It's certainly lots of fun. But having spent a lot of time working with them, with Bill, you see a lot of people there who are really dedicated to trying to do a good job under quite adverse circumstances often. And in fact, that is the backbone of American science, is the federal funding system. It has just like really broad, deep support, and it will always be where, well over 90% of the funding comes from in any good world, right? So we need to support that, but there are gaps in what they can do. It's hard for them to pick up completely new things. And sometimes something like a philanthropy or an institute can kind of pick up the gaps in what they're able to do. I think of it as the NIH is really good at not wasting the taxpayers' money. And so what you can do when you're a philanthropist is to try something that seems a little crazy, and if it works, then it can be the start of something much bigger. And if it doesn't, then it can stay where it is. And so Marian's example of kind of starting with philanthropy as she was looking at stroke and patients and then kind of moving forward from there and now having NIH funding to continue it is a great example of something that early on was like, I don't know about this. And now it's been de-risked by somebody who had confidence in you. And lots of things that happen at Stanford fall in that category from my perspective. So many people here are studying aging, and frankly, some of the, should you be studying these strange little killifish that Anne Brunet studies, or should you be studying naked mole rats that live underground, or should you be studying parabiosis, these things where you sew two mice together? I mean, these are all a little bit out there, and yet all of them have, actually in this case, they've all actually yielded insight. But doing things that might seem a little bit wild at the beginning, kind of getting those first kinds of stories going, I think is a great example of where philanthropy can have a role. And then the second kind of part of that is that different kinds of institutions have different strengths. And there will always be a role for discovery science, people doing their own thing. But one of the things that we've learned in the past few years, for example, is the value of team science. And so there's been real team science in places like the Broad Institute for Genomics or the Allen Institute for cell mapping and pathway mapping, where you really want to get a project done consistently at high quality with smart people. It's not a factory, but it's somehow, it's not a single lab. It should be done in a way that can be scaled up. And those things are largely philanthropically funded, although partly funded by the feds as well. The Genelia Connectome project. These things are really adding a lot and providing tools that are open to everyone in the community to use. And I think that's been another successful example. And then what I think of as the Stanford example, the thing that's great here and that the Institute is really playing off of is the interdisciplinary model. So there are very few places that would have the breadth and the depth and the quality, arguably no other places as Stanford, in terms of basic science at the highest level, the medical science and hospitals that can complement that at the highest level, the engineering and material science to get that kind of work done, psychology and ethics, all of these areas kind of in one place that you're trying to look at. And each of those topics has come up today. Should we be transplanting parts of the human into the rat? That's a human brain into the rat. That's an ethical question. And having Hank really involved in that is a real plus. And some of these things about nerve regeneration that Sergey was talking about, there's so many questions there that kind of come up at these intersections between different fields. And I think that this is an institution and an institute that's playing that out extremely, extremely well. Now, having said all that, how have you made sort of your decisions about how to balance? I've sort of alluded to both basic science and more clinical questions. So how have you made your decisions on what those are?
What you just said is, you just ended by saying what Stanford has here makes it both an institution but also an institute, a great place to do team science, and I think why the Big Ideas program really works so well here. But I'm just going to repeat the strengths that you just said. I mean, Stanford is a very comprehensive research university with so much academic strength and depth in so many fields, right? From engineering to business and also biology to medicine. So when you have all of those resources in such close proximity, it means that there can be more frequent interactions among these groups. And I think the chances that a really novel idea or invention that's sparked among these groups actually has some sort of breakthrough impact, I think that the chances of that go up exponentially. So this is a great place to do that. This is also a place where basic science can quickly get translated to human health benefits because of the School of Medicine. We have such a good relationship with them, and they're in close proximity. And then also, even in the technology or the measurement field, if there is some unmet need in brain or mind measurement, we can innovate technologies here in order to address them. So essentially, for me, we don't know, as we've just said, we don't know that much yet about the root causes for so many of these brain diseases that we care about. So that's why, I support basic science so fervently. But I also believe that we need to be developing more tools using technology that will help us understand the brain at a systems level and maybe allow us to understand the brain at scale, because I think the tools and the basic science will end up getting there to our end goal faster. But that said, Cori, I alluded to technology and tools, and there have been so many new tools that have come out over the last decade, like gene editing, optogenetics, advances in computational modeling. They're all very available, and I was wondering do you think there is a needed new technology right now, or is there a model on the horizon that could really help us accelerate brain research?
So it's kind of fun to realize that in 2013, when the Brain Initiative got started, CRISPR was just barely getting going, and now it's completely pervasive. It's that these tools become really, really powerful really fast. And Stanford has been a great place specifically for neuroscience tools. Carl Deisroth's work in the field of developing optogenetics, Mark Schnitzer's work in developing ways of recording many neurons. There were pictures, I think Brian showed a picture of Hubel and Weasel, who would laboriously record from one neuron at a time in the visual cortex. And started their numbering from neuron 500 to make it seem like they had done more than they really had. And now, in a single experiment, it's not that difficult to record the activity of 10,000 neurons across multiple brain regions in a living animal that might be carrying out different behavioral tasks. And it's still going up. So it turns out that at some point you become the victim of your own success, which is what are you going to do with all that data? And what are you going to do with all that information? And one of the things that's emerging from that, for example, is that, again, the sort of parcellation of brain regions into a, kind of a blob for this and a blob for that, the sort of early versions of what Brian was talking about, that doesn't quite hold up the way that we thought. And a lot of parts of the brain have what people are calling mixed selectivity, which is that you go within a certain part of the brain and you find that there's really detailed information in a subset of the cells in any given brain region about things that you thought were being addressed in completely different brain regions. That things like hunger and thirst and the nature of the reward and where you are in a task and whether you're licking or not or whether you're running around are being sent all the way back to really early sensory areas and lots of places in between. So there's this, it's, the old way was like there were these specialized regions and then they connected to others and then maybe there was some dopamine broadcast around, and now all of a sudden it's a very complex measure. And so the big problem right now really is along the lines of interpretation. It's the questions of computation and theory and artificial intelligence and data science and whatever it takes to really reach an understanding. We do have tools like optogenetics that can at least then be used to test causality, so you aren't just trying to explain a lot of noise. But I think that the really big advances are, at this point, I think people are looking toward the theory and computation groups. Again, something that's very strong here at Stanford to kind of take the next steps in the technology and to be able to interpret what we're getting out. And I have to say, I'm very optimistic about just a few things I've seen very recently, like literally, within the past year, where people are building models of brain or brain region that are inspired by the anatomy of the brain and the brain region. So you build a model of the retina that has about the same number of cell types as the retina and about the same sets of connections. And then you practice machine learning magic on it, and it comes back with something that actually looks a lot like a retina. It has all the sort of right cell types doing the right things and the right connections. And there's some similar, quite dramatic examples in the fly that are very promising toward letting us, it will be unsatisfying if we make a model of the brain that we can't understand. And so the thing that's encouraging about these new versions of machine learning and artificial intelligence is that they will let us, going through a lot of mathematics and complex data analysis, they will return the answer to us in a framework that we may be able to interpret. And I think that will be helpful. The second thing I'll say that I think that, I've been a supporter of technology development. I mean, that was something that I did both at CCI and at the Brain Initiative, thinking about what technologies needed to move forward. But I don't think technology for itself is the right way to think about it. So I think maybe the thing to do is to take a step back and say, well, what problems are we still really not solving? And I think that, an example of that is mental health. And, the thing about mental health and anxiety and depression and these things is that on the one hand, we do have some treatments that are somewhat effective for some number of people. On the other hand, we have immense unmet need for better approaches to mental health. And some of the things that have been most useful in other areas have been strangely unhelpful. So genetics has been a huge push in mental health. There have been enormous studies of depression and anxiety and what the genetic risk factors are. And unlike Alzheimer's or Parkinson's, they have been really unsatisfying. So, collectively, they account for 10% of your risk of developing some kind of a disorder. And there are now hundreds of genes that have been implicated. And all put together, they add up, they have just a fraction of the effect of what the effect of childhood trauma is on lifelong risk for depression or anxiety. So there's something very big that we don't know that is clearly related to an experience that then gets encoded in the brain in a very long-term way. And that particular powerful tool was not useful. So I'm trying to think about what is useful. Again, there are therapeutic things that are useful. We have drugs and we have direct, magnetic stimulation mechanisms and things like that work, but we really don't understand what anorexia is. If you have anorexia nervosa, you have about a 10% chance of dying. It's an incredibly horrible disorder.
But technology does help
Yeah.
us understand the brain at scale, though, right?
Yeah.
So
Yeah,
would that, for brain states and,
Yeah.
okay.
Yeah, so I think, what do we need to do? So this is the question, it's like, what do we need to do? Do we need to develop more sophisticated ways of tracking brain activity? Do we need to be tracking humans over long periods of time, or really nailing down a few of the cases where we do understand something about the triggers for these disorders? So, can we just figure out how these sort of hormonal states, like peripartum depression, work? Like, where are the hormones acting? What parts of the brain? Will that give us insight? Or how circadian rhythm affect emotional states? So can we use those kinds of things to get in? Or do we need to do, the equivalent of the study, the somologic study, where we track 7,000 proteins in human beings over long periods of time to figure out what kinds of fluctuations there are that might be telling us something about what's going on? There are hints. There's hints about inflammation in anxiety and depression. There are relationships to things like body mass index. There's something about physiology in there. But I think those are partly it's phrasing the question and then developing the technology to help to gain the answer, I think is what's called for in this situation.
But you think large data sets, AI, is a good technology that will help?
Yes.
It can be applied?
Yeah, I think, large data sets, large studies of humans, and again, machine learning studies of their brain activity, studies of the biochemistry that underlie it, studies of the brain-body axis, I think, or I have a certain amount of, I'm going to bet $20 that a big part of mental health is actually physical health, and that a lot of the things that we're learning about how the different organs are innervated by the brain and how they send information back to the brain, and how the brain affects the immune system and vice versa. Like, I'm going to put some of my money on that for some of these disorders. But I think there's still, and again, it points you toward a set of directions to ask about that I think are exciting. So it's been 10 years, and you're still here. So how do you feel about what's happened at the Wu Tsai Institute so far, and what are you hoping for in the next 10 years and after that?
Well, I think today has been pretty phenomenal. So I feel very proud. Someone also stopped me and said, you should, this is amazing. And I thought, it really is. And also that you want to be continue to be a part of this means we're also doing something right. So I think 5 years ago, I think I did an interview with Bill, and we talked about what my hopes were, and I'm going to have to say what I said was my hope then is still my hope now, which is to create new knowledge and find discoveries about the brain that will ultimately be translated into human health so that we can all live longer, healthier lives. For sure, that's there. And so let's see, what have I been impressed by in the last 10 years? Okay, number one, I have to say I'm really pleased that we have this amazing building that was constructed, and there are a lot of core facilities and labs that can now be under one roof, and so therefore collaborating and interacting. I'm proud that a lot of the SEED grant recipients and the Big Ideas grant recipients have gone on to make great progress. We know that Tony Wascore and Anne Brunet's work directly led to the Knight Initiative for Brain Resilience here. Like to see more of that. And of course, Sergio Pasca's work on brain organoids has so much promise. Those things germinated here, and I'm very proud of that. Third, happy that scholars have gone out and participated, and then some of the students that we heard today. They're at different universities, but clearly they embody so much of the interdisciplinary mindset, that is part of Stanford, things that germinated while they were here, and that's something to be proud of. you just mentioned, I love the fact that we have computational, theoretical, and experimental approaches to understanding the mind and brain right here, and that's clearly going to be a part of our bright future. So I'm glad that that's already established. And I think, Jill, where are you? You'll be happy to know that I think that vibrant community, you have succeeded on that one. So I'm happy with that culture that started with Bill and that continues to be carried on. So as far as the future, well, I'm going to say, I still want to find cures and biomarkers for neurodegenerative diseases, but for everything else, I'm a +1 to everything you just said. But for me, that I'm excited about our neuroscience theorists working with large data sets and AI, because I think it's through that we're really going to be able to understand how cells and molecules in the brain actually coordinate their activity to compose human behaviors. And I think understanding the mind is really critical to understanding and creating the future society that we want to live in. So, this ultimate aspirational goal of understanding the mind in all of its complexity is still the right one, and hopefully in the next 10 years we're just going to come closer and closer to reaching it.
Well, Stanford is lucky to have you as a friend.
We're lucky to have you as a friend. Hug.
Thank you so much. I genuinely have learned a lot. And as a gesture for our gratitude, we have small gifts. This one is for Cori, is a book about art and brain. And this one is for Clara, is A Brain on Fire.
Okay, great.
Oh, that's a good book.
Oh, really?
Yeah,
Good,
yeah,
good.
yeah.
Thanks. Did we do good on time? If there's more, we should ask questions to Cori.
Oh, thank you. We didn't plan for the questions. If there are any burning questions, maybe we'll allow one or two questions.
Headline news in the last 6 months has been about OpenAI, ChatGPT, huge multi-layer architecture. Now, very simple neuron models. What should they add to make them smarter? Here, they need a nuclear power plant connected to a data center. We've got peanut butter and jelly sandwiches connected to a 1.5 pound brain. What aspect of neurobiology would you add to those models as low-hanging fruit to make them better?
Yeah, interestingly, some of the more engineering-oriented people, as they try to develop systems that can do what the brain does, become more and more impressed by how the brain uses very few calories and generates very little heat and still can solve all of these problems to do it, right? So evolution is a very strict teacher. And so it's obviously pruning out all of the possibilities to generate something that works very well. And I think maybe that's a different way of saying that the idea of using the brain to inspire some of the AI is another way of making those methods more efficient. And I like this example for obvious, the very first artificial intelligence type methods that were developed, the ImageNet methods, were developed by Jeff Hinton, who was a theoretical neuroscientist. And he developed image analysis so that you could recognize your friend's face on Facebook or whatever, or it would recognize it, based on what he knew about how area V4 recognized different kinds of textures and shapes. So it was a brain-inspired mechanism that led to that first major advance in AI. And so I think that that's something maybe that this can, under the best of circumstances, be a virtuous cycle. And another part of that, and I think I should be reluctant to talk about this kind of tech and AI here at Stanford, given how much more people here know about this than I do, but that doesn't usually stop me, so it's not going to stop me now. And I think that one of the things that we're seeing is, right now the kinds of tools that people are using, they're trained on everything. All possibilities, all possible outcomes. But you can make something much tighter by training it on high quality information or highly relevant information. And so there are ways of building these things in simpler ways that are now being, training language models on the scientific literature will probably give you higher quality answers to scientific questions than training large language models on every word ever published on the internet, right? So I think that those are mechanisms, again, of sort of using some smart filtering to at least try some different possibilities for how to simplify these and improve them.
Okay, we'll take the last question.
You mentioned that one of the initiatives that you're really proud of is with brain resilience, and I'm just trying to get your comments on, just as the public thinks about heart health, I think there's not as much understanding of like really what brain health is, what is like a checkup for brain health. So what do you think is the long-term vision of partnering with epidemiology, environmental scientists to really make brain health for all a reality?
So who's that question for? I think it was Clara who talked about the resilience being the part that she was really proud of. And I think this is something where, what the Tsai had built was able to capture someone else's attention and curiosity and bring them in. And I think that's a great model in general, so that each place can kind of keep a character, but then a new piece can grow onto it. I think that this is some of these questions around resilience and mental health. It's going to take the equivalent of the big ideas. It's not going to come from top down from me or from Clara or from Kang deciding that we know how brain resilience works. It's going to come from someone who's willing to take some risks and a lot of smart people who are willing to generate some ideas and carry them out to see where those go. And that filtering process, I think, is so I think that the big ideas were bottom-up ideas, is my understanding. So
Very much so. Yeah.
So that seems to work. So it seems worth. Is that okay? Yeah. Okay.
Thank you, Cori and Clara, again.